Sample size calculation for oral microbiome studies. Sweki. In this short video, I explain how to calculate sample size for oral microbiome studies based on the different diversity measures. I have calculated sample sizes from the information derived from published studies. You can access the studies by scanning the accompanying QR code. Pause video when you want to scan the QR code. I've explained a little about sample size and the parameters. It is suggested to look for other complete resources for a full understanding of what is sample size. About me, I am Shaju, professor in Rigas Stradiņa Universitate, Latvia. Human microbiome studies evaluates the relationship between microbiome features and a biological or clinical condition. It can be also an environmental exposure or medical intervention. Earlier, not much was known about microbiome, and many data sets which were available were huge and were not normally distributed. Now, as more evidence and understanding happens, this research lends itself well to hypothesis comparison and testing. So, there is a need to understand the basis and methods of sample size calculation in oral microbiome studies for the effective and efficient use of resources towards a robust evidence. How to identify resources that can give you the parameters for sample size calculation. Similarity of microbiome analysis. 16S rRNA based bacterial community profiling via next generation sequencing is currently the standard procedure to determine the composition of a complex bacterial communities like a oral microbiome. 16S rRNA gene based analysis, which are particularly effective for the characterization of the oral microbiota thanks to the highly curated databases and extensive literature available. Second is data summarization. The number of sequences that correspond to bacterial taxa are operational taxonomical units or OTUs. OTUs are synonymous to bacterial species, except that different related species can have the same OTUs. The OTUs are typically presented as relative abundance than absolute abundance. To overcome this came the newer one, amplicon sequence variant. It is more advanced and sensitive than OTU counts and detect differences among sequences by much as a single nucleotide difference. Select studies for sample size which are as close as possible. For example, if the study design is observational, go for observational references. Cross-sectional versus cohort studies. Select studies that have used similar methods for sample collection. Now, the most popular method for oral microbiome appears to be simple saliva collection, which has shown good validity for oral microbiome characterization and association. A little explanation of statistical concepts relevant to sample size calculation. Again, for a full review, seek relevant resources. Type 1 error or alpha error is the probability of rejecting the null hypothesis when it is actually true. When two groups being compared are similar and the results show that they are different, then that error is alpha. It should be low and less than 5% or 0.505 is acceptable for clinical studies. Type 2 error or beta error is failure to reject a false null hypothesis, and it is the opposite of alpha error. 0.2 is a reasonable error for a clinically reliable study. Power of a study is a complement of beta error. 1 minus beta is power, which shows how reliable the study is. It is kept at 0.8 or 80%. Effect size is the difference in the outcome variable between the two groups. It can be measured as mean and standard deviation, median and interquartile range, odds ratio, proportions, etc. When the effect size difference, example mean difference is small, we need a large sample. If the variation in a group is high, then we need a large sample. So these are the 4 parameters for calculating sample size. There are complex equations that calculate sample size based on the type of effect. OpenAP is a reliable, simple sample size calculator that is ideal for beginners. Let's understand effect size in microbial context. There are few ways of effect sizes based on the measures. The first two, species richness and the presence or absence of a species, are simple measures and are not discussed further. Species richness, the first point, refers to the number of different species in a sample. It is a continuous measure. Standardized mean difference is the effect size. Second, when the effect measure is the presence or absence of a species, then odds ratio is a suitable measure for effect size. Third and fourth are the popular measures for effect sizes in microbiome studies. Third, alpha diversity is within a sample, accounts for the richness and evenness of the species. Beta diversity is across samples and tells us how diverse are they. Alpha and beta diversity and sample size calculation with them as effect sizes explained in detail in the next few slides. Beta diversity. Beta diversity assesses the diversity among different samples. Diversity between a microbiome of patient A and B in a group. Bray-Curtis measures how different the species composition or the abundances are between two samples. Jacquard index focuses on shared species, whether they present or absent between two samples. Unifrac or unique fractions is a dissimilarity is based on phylogenetic relatedness of the OTUs. I'm using unifrac values from a study by Zakari et al. For calculating sample size for a similar study in a different population. The values of unifrac ranges from 0 to 1. Two groups, adults and youth, are compared in the referred study. Please scan the QR code for getting to the referred study. The figure 1D is a box plot comparing the groups by medians and interquartile range IQR. This values, the medians and interquartile range, was used and converted to mean and ST and used in OpenAP. So we got a sample size of 393 per group. Alpha diversity. Alpha diversity refers to the number of species present in an ecosystem, richness, and frequency of occurrence of each type of the organisms, evenness. More diversity in a sample is indicative of health. Evenness is the similarity of the abundances of different species. Shannon and Simpson are popular indexes for evenness. Shannon is affected by rare species, while Simpson is affected by the proportional abundance of the most common species. Faith, PD, or faith phylogenetic diversity, is a popular metric for alpha diversity, which accounts for the phylogenetic relatedness in calculating the diversity among the different species in a sample. Alpha diversity for sample size. Here I provide an example of sample size calculation using the difference by faith PD metrics. Faith metric is given as the sum length of the phylogenetic tree branch. Scan the QR code for the referred study to take the parameters. The tertile 1 and 3 groups are compared and the parameters are given. The sample size is 2420 as the mean difference is small between the two groups, while the tertile has a large standard deviation. Thank you. Post your comments, corrections, and areas for improvements. References and explanations are provided in the description below. Thank you again.